Presented by Caltech.
Okay. All right, so good afternoon, everybody. First, I want to thank the organizers and the committee for giving me the opportunity to present my work here. I'm actually really, really excited about being here. And so today I want to give you a brief overview of the general questions that I'm interested in. Then I'm going to talk about the work that I did during my postdoc with Cliff Tabin in Boston. And then in the last 10 minutes or so, I really want to talk about what are the future directions that I want to take, what are the questions that I think that are important to be addressed in the next and coming years. And so more generally, I'm sure like everybody else who took a biology class, I'm really interested and fascinated by this beautiful diversity that we see out there. I'm a little bit too loud, I think. That we see out there in nature. And more specifically, I'm really interested in what is the genetic basis. I mean, I'm a geneticist, I'm a developmental biologist by training, so I'm really interested in what are the genetic and developmental mechanisms underlying this variation. But also something which I believe is too often forgotten, but I think it's fundamentally important. I mean, I'm sure you've seen diversity slides like that very often, beginnings of talks, but what is often missing, but is really tightly linked to this beautiful diversity that we see in different animals, is the equal diversity that we see in different habitats, different environments, different climates, different ecological niches. And obviously the reason that these two are so tightly linked is because animals have always been able to adapt to these different and changing environments. And this is the reason we can actually admire this beautiful diversity that we see out there in nature. So I'm deeply convinced that if you want to fully understand this diversity in different animals, including as humans, it is really important to keep the environment, the selective pressure that was acting during adaptation in mind. And at the very end of the talk, you will see why I think this is specifically important also for human evolution. The particular environment that I'm using to study adaptation is the cave environment, which is a rather extreme environment. But even to such an extreme environment, animals have always been able to adapt. And cave adaptations include losing their eyes, losing their pigmentation, but many, many other traits as well, where the adaptive value is much clearer and also very, very striking and interesting. I'm going to come to that a little bit later. And the particular model system I'm using to study adaptation to this extreme environment is the cavefish Astyanax mexicanus. And so for those of you who are not familiar with the system, that's why I want to briefly introduce to you the system. So these cavefish, they live in this little area here in northeastern Mexico, in the Sierra de El Abra. And basically, so these are mountain ranges, and every little circle represents a cave that has its own cavefish population. And cavefish populations are named after the cave they are found in. So for example, Tinaja, Pachan, or Molino. You will hear these words more often during the talk. And so this is how they look. So they come in two forms. So the ancestral, the derived, the surface form that you can, the ancestral river form that you can still find in these rivers nearby, and the derived cave forms. These are just three examples of different cave populations, the one that I just mentioned before, Pachan, Molino, and Tinaja. And I think, and this picture already brings out an important point. So many of these cave populations, specifically the ones that are depicted here, have been invaded independently. So they represent independent experiments of evolution in nature, always with a very similar outcome. So it's a great system to study convergent evolution, parallelism on a population, on a whole genome level. The system has a couple of other key advantages. Probably the most important one is that they are still the same species. So all four fish that you see here are Astyanax mexicanus. So they're completely interfertile. You can cross a surface fish to a cave fish. You get fertile progeny, which allows you to do genetic studies, QTL analysis, but also developmental studies, complementation tests, and so on. More from a technical perspective, these cave fish live and breed in the lab. In this respect, they're actually very similar to zebrafish. And last but not least, we're dealing with a relatively clear and very, very strong selective pressure. So we have a very good understanding and idea about the environments these fish live in and what the selective pressure are. So for example, these are the river environments, beautiful rivers surrounding these caves. It's gorgeous there. You can swim with the fish. But it's a classical river environment. There's light, there are predators, there's food. On the other hand, the cave environment looks very, very different. It basically looks like this. So it's pitch dark. But the good thing is you can actually turn on the light, and then you can see these fish, you can measure parameters. They will be relatively stable over the course of the year. And so this is always, so we have a good understanding of this environment. And this is always the picture that I show because it nicely depicts the cave fish. But just to give you a little bit of an idea how it actually is to go into these caves. So these caves are usually covered with bad guano and mud. You have to crawl through that. You have to crawl through bugs in the entrance of the caves. It's not a very pleasant environment. You also have to wear a mask all the time because of this. In this bad guano, there's a fungus called histoplasmosis, which can infect your lungs. Because of this mask and the high humidity, it feels really, really hot. So this is me maybe after 20 minutes. This is me after an hour. So you see the color of the shirt has changed. And so it's really not a very pleasant environment. But eventually you reach your cave fish, and this is kind of your Darwin moment when you see the fish you're working with and you're really excited. You do all these experiments until you realize, man, I have to go back this entire way. But anyway, we do this from time to time, but we don't have to do this all the time because they live in the lab. But we have a good understanding of the environment. And so taking advantage of these key characteristics of the system, over the last couple of years, I've been working on several studies using these fish. So we, for example, mapped some behaviors, some feeding behaviors. So these cave fish, they feed in a very different angle than surface fish. So they feed in a kind of a 45-degree angle, scanning the ground, while surface fish feed in a 90-degree, very steep angle. And this is actually genetic. So we could map this trait. We found genetic loci that are linked to that. We mapped some morphological traits that could potentially explain this. We looked at the loss of schooling behavior. So schooling behavior is important if you want to hunt together, if you have a lot of food, if you want to avoid predators. All this is no longer necessary in the cave. And so we found that this is due to site-dependent and vision-dependent and vision-independent genetic loci. We also have now a cave fish genome. I was part of this. I'm really, really excited about this. This is another big advantage of the system, that we do have the genome now. So I think this is going to change the field drastically. But today I'm going to talk about a different study which deals with the role of cryptic variation in these cave fish. And so this is technically a systems developmental biology position. So and this particular study deals with a very classic concept in systems biology. So I'm going to come to that in a minute. But this study deals with the question of where do these mutations that play a role in evolution and adaptation, where do they come from? Do they come from de novo mutation or standing genetic variation? And by de novo mutation, I mean you go into a new environment, let's say in this case the cave environment, and then you acquire a mutation. If this mutation helps you in survival, will eventually get selected for or fixed in the population. On the other hand, standing genetic variation means you go into a new environment and you already have a pool of mutation from which basically nature can select out the ones that help you in survival. And both are very classical concepts. So they're valid concepts. There are examples for both of them. But I think it has become more and more clear over the last couple of years that standing genetic variation is really an important contributor to evolution, to adaptation, especially if you're dealing with quickly changing environments, because it's just so much faster than de novo mutation. However, it does have an alternate problem. And this is, how is it actually possible that you can accumulate these mutations in the first place? Because technically, these mutations that give you an advantage in a new environment shouldn't be advantages in the old environment. And this brings this classic concept of canalization into play. Canalization is a term that was coined more than 70 years ago by Conrad Weddington, one of the fathers of systems biology. And by canalization, he basically meant that there are certain phenotypes during development that are more or less robust against slight genetic variation. So if you imagine development going down this path here, you will always end up with the same phenotype independent of slight genetic variation. And only if you have a very strong event, a decanalizing event, and Weddington could show that this could be, for example, an environmental stress, like a heat shock, then you become decanalized, and you end up with another phenotype. So this variation becomes, genetic variation becomes visible, and you end up with another phenotype. And he beautifully showed in fly experiment that such a mechanisms exist. But he actually put it a step further and said that such a mechanism could actually play a role in evolution. It's kind of a paradox. So how can a mechanism that prevents variation, how can this be involved in evolution? But this brings us exactly back to our standing genetic variation question. Because of such a mechanism, you can actually accumulate and maintain these mutations in the first place. And then let's say you go into a new environment, which is very stressful. This genetic variation becomes visible, you become decanalized and can be selected for. Beautiful theory, very controversial. First of all, he didn't really know what such a mechanism could be. And it took almost 50 years until Susan Lindquist found out that HSP90 is such a canalization mechanism, probably the main canalization mechanism, especially for the phenotypes that Weddington described. And HSP90 is actually also this perfect protein for this role in evolution because it has this dual role. It has this canalization function of keeping variation quiet, but also probably the better known function of being a heat shock protein, a protein during environmental stress, which keeps kinase proteins from misfolding. And so you could really imagine that you go into a new environment, which is very stressful. HSP90 is busy with its heat shock function or its stress function, and therefore its canalization function is deprived, and you can get phenotypes that could be technically selected for. And this has been shown beautifully in fly experiments, in their laboratory experiments, basically, but it has never been really shown in a natural system, and especially not in morphological traits that you could technically select for that are important, potentially important. And so that's why we set out to answer the question if canalization through HSP90 maybe played a role in cavefish evolution. Why cavefish evolution? Because we believe that if such a mechanism exists, takes place, it was most likely take place in a setting where members of a species are very suddenly confronted with a completely new set of environment. And this is exactly the case in cavefish evolution. All right, so we basically ask two questions. So do natural populations of surface fish actually carry such standing genetic variation that is released by or that is controlled by HSP90? So if we block HSP90, do we get an increase in the variation in morphological traits? Has never been really shown before. And maybe more importantly, what would be the situation in cavefish? Because if such a mechanism took place in cavefish evolution, you would see a differential response. There shouldn't be an increase in the variation anymore. There should be some alleles would have been selected for, so there should be a differential response. All right, so for the first question, we used natural populations of fish, these surface fish. And we used the chemical inhibitor, which is called radicicol. It's widely used in the field. It's very specific. It blocks the ATP binding domain of HSP90, which is highly conserved. And it basically mimics an environmental stress. And when we use this chemical inhibitor, what we do see is an increase in the variation in eye size, and eye size being an important trait in evolution, in cavefish evolution. So and a trait, a really morphological trait that could be selected for. And so we do see this increase. So the mean is not changed, but the variation is increased. So we get larger eyes, smaller eyes, and so on. And so this has never been really shown before, and is very exciting. And this is we did this in adults, but you see that already during early development. So it's something that happens also already during early development. We don't know exactly when it's happening, but you can see that already in larvae. But these experiments, most of the experiments were done in the adults. All right, so that's interesting. But what is the situation in cavefish? So if we repeat these same experiments, same species, but different populations, we get a completely different result. We don't get an increase in the variation anymore, but what we do get is a further decrease in eye size. So in this case, we couldn't look at eye size anymore because cavefish don't have an eye anymore, but we looked at orbit size. They still have an orbit. And it's the same for surface fish. You can do that with the orbit size as well. And so we do get basically cavefish that have smaller eyes. And this basically tells us that the alleles that have been selected for in cave evolution are at least alleles that are responsive to HSP90. So only the ones that give you smaller eyes are still there. The other ones are gone. And so this basically makes an involvement of HSP90 in cave evolution at least plausible. And so, but this is basically just transient, so it's a chemical inhibitor. So can you actually select for this? And so when we repeat these experiments with surface fish, and now take the ones that have smaller eyes after the inhibitor as being an adult, and then incross these in the next generation, already in the next generation, without the chemical inhibitor, you get fish that have smaller eyes. And it's something that is called genetic assimilation, has been shown in flies. It's nothing very spectacular, but it basically tells us that what we are seeing is actually really genetic, and it's not the inhibitor messing up the development of the eyes. All right, so this was cool, but as I told you, this has never been really shown in a natural system, so we also don't know what could be such an environmental setting. What could be such an environmental stress? I mean, evolution usually has these gradual changes, and it's not that you had a particularly hot summer and then you get a new horse. So what is the actual environmental stress? But as I said, the good thing is we can actually go there and measure parameters. And we did this, and despite the fact that it feels very hot and humid in these caves, it's actually not, the temperature is not different in between the surface and the cave environments, at least when we were there. But what was different is the conductivity. So these caves have a much lower conductivity. And so conductivity is basically the dissolved salt content. And obviously, that's an important stress factor for a fish. So if you take an ocean fish and put it into fresh water, it's not going to like it. And it's also been shown that this goes actually through HSP90, so that HSP90 is also activated by osmotic stress. So this was a very good candidate to look at. And so when we repeat these experiments now, without the chemical inhibitor, but using this lower conductivity as an environmental stress, we do get basically the same results. We do get an increase in the variation, again, in both directions. It's not as extreme as with the chemical inhibitor, arguing there might be other stress factors as well playing a role. But at least it's the first time that we can really link an environmental stress with such a observation. All right. So what I showed you so far is that for the first time that has been shown in a natural system that HSP90 could play a role in evolution and in really linking an environmental stress to this phenotype. All right, so for the second part of my talk, I'm going to go a little bit away from cavefish. I'm going to ask the question of what are actually these types of mutations. So what I showed you are that these mutations, where these mutations can come from, but what are actually these types of mutations? They can be either coding mutations or regulatory mutations. And for this particular project, we decided to focus on regulatory mutations, and we actually looked at human evolution. Why did we look at human evolution for this? It basically goes back to this very famous paper from Mary Claire King and Alan Wilson almost exactly 40 years ago. And what they found is that if they compare human and chimp proteins on the amino acid level, they're actually very, very similar. So similar that they cannot, at least they say, not account for these vast biological differences that you can observe. And so they argue already at the time that it must be regulatory mutations that drive human evolution. And 40 years ago, this was a very, very bold statement, but I think nowadays it's clear and widely accepted that regulatory changes are very important during evolution, and maybe specifically for human evolution. So for this particular project, we were really interested in these types of mutations and in these phenotypes, these human-specific phenotypes. These are just a couple of examples of human-specific traits that you can see if you, for example, compare us to our closest living relative, the chimpanzee. And despite the fact that these are very important traits, it's really surprising how little is known about the genetic basis of these traits. And this is even more surprising given that we do have the human genome now since 13, 14 years, the chimp genome since more than 9 years, and many, many other mammal and vertebrate genomes as well. I had actually the pleasure to contribute to two of these, the coelacanth and the sea lamprey. And I think this is a very valid resource, and I think now we have roughly 70 published vertebrate genomes. But despite all this wealth of information, it's really surprising how little we know about the genetic basis of these traits. And so that's why we wanted to take advantage of these genomes and, as I said, look specifically for regulatory mutation. So what did we do? We basically aligned human, chimp, macaque, and mouse genomes, because these are very well annotated genomes, and then looked for regions that are more or less conserved between these species, potentially indicating some regulatory elements. And then we looked for human-specific changes in these regions. Obviously, there are a lot of these, so we looked for very, very drastic changes. We basically looked for deletions that are larger than 100 base pairs that are unique to the human genome and that are regions that are conserved in these other species. And so this was done in collaboration with Michael Zodi from the Broad Institute. He's really a brilliant bioinformatician. And so together we came up with a list of roughly 300 human-specific deletions. And all of these deletions were actually in intronic or intergenic regions, so could potentially remove regulatory elements. And these are just a couple of numbers of ones that are nearby a gene. But they could also be just some random deletions in the genome, so we had to test this. And how did we test this? So we basically used the transient transgenic approach, where we take the piece, the corresponding piece that is missing in human, take that from the mouse, put it back into the mouse, but this time in front of a minimal promoter, which would only drive like Z expression if this piece has some enhancing some regulatory function. And then you screen the progeny for tissue-specific expression in structures that could be interesting for evolution. And so we did this with 12 different deletions. We also used 2 empty controls just to be sure that what we're seeing is specific. And out of these 12, 4 showed very tissue-specific expression in very interesting structures. So for example, one is expressed in the pons, the other one the intercostal muscles, so these could maybe play a role in breathing control. But we didn't focus on these ones because we have more expertise in bones, we have more of a bone lab, so we were focusing on the ones on the right, and these are the ones I'm going to talk to you today. And so the first one is a deletion that is downstream of a gene called odd-script related 2, which is a regulator of osteoblast differentiation, and upstream of a gene called VPS13B. But the reason that I believe it's regulating this gene is if you look at its expression. So on top is wild-type expression of this gene, and on the bottom is the transient transgenic expression that we see. And in every structure that we see expression, this gene is actually also expressed. But this gene is expressed in other structures as well, so it really looks like we just have an enhancer, kind of a subset of its expression, really like a regulatory element. And these are actually very interesting structures in terms of human evolution. So it's expressed in the palate. We do have a differently sliced, differently shaped palate. It's expressed in the Meckel's cartilage, which plays a role in lower jaw development. It's expressed in the basioccipital bone, which is shorter in humans, and one of the reasons that the foramen magnum sits in the middle of the skull and the skull on top of our body, and we can actually walk upright. So that's interesting, but how do we test this? So if you look at the full knockout of this gene, this is not really helpful. It's embryonic lethal. It does have a palate phenotype, but that's about it. So for this one, we decided to mimic the situation that we see in human in the mouse by just removing the piece that is missing in human from the mouse. And when we did this, so these are embryos that have this deletion. And so basically what I did was a very crude assay just to take the head of the embryo and to see if its expression is changed of odd-script related 2. And this is just quantitative real-time PCR. You see it's about a 2-fold downregulated, which is not a lot. But as I said, this was just the entire head, so we have to do section in situ and to see if it's a general downregulation or if it's really certain substructures missing its expression. But basically, the take-home message of this slide is just that we're looking at the right gene, because if we look at the other gene, its expression is unchanged. All right, so what's the phenotype? That's obviously an important question. And when we started this project, we were maybe hoping for a very drastic phenotype, like a walking mouse or something like that. But obviously, this is never going to happen. And I think it has become more and more clear in the field of regulatory elements that if you knock these out, you're not going to get very, very drastic phenotypes. There's always some redundancy. It seems like they're more fine-tuning development. And so the same is true here. So these mice look like mice, and they are viable. So what we have to do is we look at the skull, and we're going to do CT scans of the skull and principal component analysis to see if there's some change in the skull. But you can maybe already appreciate that it looks like the snout is a little bit shorter, the positioning of the vertebrae a little bit different. But we really have to look at many of these mice and do principal component analysis to be really sure that this is the case. But very quickly, I'm going to talk about the second one, which I think might be even more interesting. And this is a deletion that is in the middle of the first intron of a gene called activin receptor 2A. It's nicely conserved. And the expression that I get is very specific to the primordium of the upper incisor tooth. So every embryo that I slice has this very specific and peculiar expression in the primordium of the upper incisor tooth. And that's actually very interesting in terms of human evolution, because we do have much smaller incisors than, let's say, chimpanzees. And interestingly, if you completely knock out this gene, so in a full knockout of activin receptor 2A, you don't have incisors at all. So you could imagine that if you completely lose this gene, you don't have incisors. If you lose a little bit of its expression, you have smaller incisors. And so for this one, we also did this knockout, this human-specific knockout, but we don't have homozygous yet. So we just have the first transmitted. And so we will see pretty soon if this is actually going to work out. All right, so for the last 15 minutes or so, I'm going to talk about what I'm going to go back to cavefish and what I want to work in the future, because I think the cavefish right now is a very good system to take advantage of to answer these questions, because now that we have a genome, that we can do functional analysis, I think this is going to be very exciting. And to do this, I want to go back to the slide that I showed you before, because I think it very nicely highlights the fact that despite they are the same species, they look actually very different. And as a matter of fact, these cavefish differ from their surface fish counterparts in a variety of different morphological, physiological, behavioral traits. And these are some of them. The adaptive value is very interesting and striking, like they have an increase in neuromass number, they have an increase in taste bud numbers, they have a better sense of smell, they have all sorts of different behaviors. So I'm convinced there would be a lot of potential for collaborations, but I'm really interested in one particular phenotype, because I think it's one of the most impressive physiological adaptations that you can imagine, and this is this metabolism that you can see in these cavefish. And so what I showed you so far is, what I told you in the beginning is that it's completely dark in these caves. So there's no photosynthesis, no primary producers. So basically there is no food in these caves, absolutely nothing. So cavefish completely rely on food that is coming from the outside. And this can be either bats. However, some of these caves are relatively deep, so they are not bats anymore. Or it could be seasonal floodings. And these seasonal floodings happen basically only once a year. And so what we think is happening is that these cavefish eat only once a year and then starve for the rest of the year. And so these are pictures that have been taken. So we go there usually right before the flood comes in. I mean, not right before, maybe 2 months or so before the flood comes in. But these fish, it's a little bit hard to see on these pictures, but these cavefish are really skinny. And so we really believe that they only eat once a year. And there's some anecdotal evidence where they starved cavefish for 9 months. And even after 9 months, they had higher fat contents than surface fish that were fed every day. And so we found this is really interesting. And so we wanted to study that. And so first, we repeated these experiments, not with 9 months, but already after 2 months, you can see that that cavefish are much better in starving than, so they lose less weight during a starvation than surface fish. And there could be a variety of reasons for that, but probably the biggest reason is that they're actually very fat to begin with, and fat is the most efficient way of storing your energy. If you've ever tried a diet, obviously you know that's the case. And as a matter of fact, if you look, these cavefish are really fat. So this is some belly fat here, and they have even these huge lipid cells full of fat, and it seems like they have different lipids in these and in these larger cells than compared to the smaller cells. But if you look at the entire triglyceride content, you see it's really drastic. So these cavefish are really fat. And there seems to be even some population differences between these caves. This is even more striking if you look at the organ level. So these are livers of these tinaja. These tinaja livers are really, really large compared to other cave and the surface livers. And if you stain for the fat content, these are really, really fatty livers. This is basically fish foie gras. And so we're really, really interested in how do you get so fat. And one way, if you get so fat, obviously, if you eat a lot. And so we measured the amount these fish were eating this is over 24 hours, but you see basically the same result if you do that over a week. So these cave fish, at least these tinaja population, they're constantly hungry. And so this is really interesting. And so what is the genetic basis of this? And so we're doing RNA-seq, we're doing QTL analysis to find this out. But in the meantime, because we do have a genome, we can actually look at some candidate genes for coding changes, for regulatory changes. And what we did find is actually a very interesting mutation in an amino acid that is highly conserved in all other vertebrate species in a gene called MC4R. I'm sure you're familiar with MC4R. It's actually a very good candidate. If you knock this gene out in mice, you get mice that are constantly hungry, that are overeating, that are really, really fat. But also in humans, MC4R is the most monogenic cause of human obesity. And actually, exactly the same amino acid that is mutated in this one cavefish population is also mutated in a case of human obesity. And in this particular case, they tested in vitro they showed in vitro that this leads to reduction of MC4R. And so when we repeat these experiments in vitro with our tinaja allele versus the surface allele, we do see also a similar reduction in the activity of this gene. But this is in vitro, so we were also interested, can we do this in vivo? And we were a little bit lucky that, because I told you, so these pachon fish do not have these mutations, but this other cave, this pachon cave. But what we do find is that some pachon cave fish have actually this mutation. And so when we now compare only pachon cave fish that have this mutation versus pachon cave fish that do not have this mutation, everything else being equal, we do see this increase in the appetite in these ones that carry this mutation. It's not explaining the entire increase in the appetite that we see in the tinaja population, so arguing there might be other genes involved as well, but this is just basically one gene contributing to it. And I told you that one advantage of the system is that we can look in different populations. And so when we look at other cave populations, so I told you the pachon fish, we don't know yet what the actual situation in nature is, if they carry this mutation or not, because some of them do and some don't. But in all other caves that we looked, they carry exactly the same mutation. And this really strongly argues for another case of standing genetic variation, because it's very unlikely that you would get the same exact mutation over and over again in different caves. And as a matter of fact, if we look in surface fish populations, so we looked in over 100 surface fish, we do find this mutation being heterozygous for maybe 5% of the fish are heterozygous for this mutation. And these are numbers that are consistent with standing genetic variation. So this is basically another example of standing genetic variation. But I told you it's only one gene, and we are looking really interested in all these other genes that are playing a role in this. And so what we're doing is right now, this is just work that has been already started. It might be already finished when I start. But these are just examples of work that you can do with these fish. And so basically what we're doing is RNA-seq of the livers, comparing cave versus surface and starved versus fed. But you can basically imagine doing this with the brain, with the hypothalamus, and so on. But this is just RNA-seq. RNA-seq is not going to give us the actual genetic basis. So the advantage of the system is that we can do QTL analysis. And so we can really map these traits in these cave fish. And we are in the comfortable situation of having all hybrids of all different possibilities of these three different cave populations. And I think there are not many other labs out there that have this. I think we're the only ones that have really all hybrids of these. And I can take this with me. So this is really a wonderful resource. And then we can map all different traits in these fish. And then because we have the genome now, we can actually find the genetic basis. And once we have the genetic basis, we can functionally test these in the fish, and this is something that I think is really missing in many other evolutionary model systems. But it works. We can now use CRISPR-Cas9 in cavefish itself. It does work. We're in the process of doing this. It's obviously a little bit more challenging than doing this in a zebrafish, but we can also use the zebrafish to screen through a lot of these changes and see which one are the most important one and test them in the cavefish. All right, so these kind of short-term projects. But in the long term, I'm really interested in how are these cavefish actually dealing with the higher fat content? Because as a matter of fact, they are very healthy. They live as long as the surface fish. They breed, and they don't show any sign of distress. And this is even more surprising given if you look at their blood glucose levels, for example. So these are fish that have been starved over 2 weeks. So this is the feeding point. And so surface fish, there's a little bit of variation in blood glucose level right after feeding, but then it stabilizes at values that you and I have, that every other mammal or fish have. It's very constant blood glucose levels. However, cavefish, when you feed them, they become super, super hyperglycemic, and then they drop drastically, and already after 2 weeks, we cannot detect the blood glucose anymore. So these are beyond the detection of the assay that we're using. So basically, these are diabetic fish. These strong fluctuations are really crazy, but they don't care. For example, if you IP inject glucose into their blood, at these high values, these surface fish go into a glucotic shock. They go basically into a coma. They don't move until they very quickly are able to empty their blood from the glucose again and get the normal levels back. While cavefish at these levels, they don't care. They swim around, they are happy, and then they're also not very keen in cleaning out the blood glucose. And so this is really interesting. And we also found out that they have much lower insulin levels, that these surface fish, and these are just, for example, antibody against insulin, it looks like they have less islet cells as well. And so, but these are really mappable traits, and so we can find a genetic basis of how do they deal with these fluctuating glucose levels, insulin levels. And I think eventually this will help us in helping more about our own disease, because if you are diabetic and you take insulin, it helps, but it's not always going to be perfect all the time. So finding ways how you can actually deal with this would be very helpful for our own health. But I think specifically this phenotype of these high fat content in these cavefish is an excellent phenotype to study human evolution. And the reason why this is because we as a species, from an evolutionary perspective, so everybody of us is fat. And this becomes maybe very clear if you look at this picture. So no one would ever say that this woman is fat. Obviously, it's the contrary. She's way too skinny. But if you look at the body fat and compare this with other mammals, most other mammals, maybe except whales or so, they have a much lower body fat content than humans. And this becomes specifically clear if you look at the body fat of newborns. So even newborns of hunter-gatherers have very, very high body fat levels compared to, let's say, chimpanzees. And I mean, that's a good thing. There's a reason why we became so fat. People argue that it might be because it helps us to have a larger brain. But actually, we don't really know why that's the case. There are other theories that people say it's because we were wading through water all the time. And I mean, that's a real theory. I mean, I don't think that's true, but it's really, I've seen a post on a scientific meeting. And so but just basically argues that we don't know why we became so fat. And so having a system where we really do know the adaptive value or the selective pressure that was acting will eventually help us in understanding more about our own evolution and maybe also about our disease susceptibility. Because this is just not only true for diabetes or eating disorder and so on. So this is a list of human-specific diseases that we only have because we are not adapted to the environment we're living in. So these are diseases that hunter-gatherers do not get. And so understanding more about the adaptive and the selective value will eventually help us. I'm really convinced about our own evolutionary history. All right, with that, I want to thank Cliff, being a fantastic mentor. The work was done in collaboration, the work on HSP90 was done in collaboration with Susan Lindquist. I had the great pleasure and opportunity to work with two brilliant graduate students, Joanna Kowalko on the behavioral studies, Ariel together now on the metabolic. We have very nice collaborators in the cavefish field. We do have some experts on human evolution on board. Some funding. I also want to thank my graduate supervisor, Christiane Nussen-Vollert. I didn't have time to talk about the work that I did in her lab, but it's all published. But last but not least, I really want to also thank some summer students that I had over the last couple of years. We have a wonderful program in Harvard which is for Native Americans. So these are all Native American students. And so this was a really great opportunity for them, but especially for me to understand questions that can come up at an undergrad level. And so that was really great. And with that, I want to thank you, and I'm happy to take any questions. Exactly 40 minutes, wow.
In your, when you were looking at human deletions and then translating them to the mouse,
yes.
I couldn't help but notice that a lot of the ones where you got a phenotype were in the neural crest. And do you think that's significant because you had Meckel's cartilage?
Yeah, no, it's true. Yes. Yeah, so if she noticed that a lot of the deletions that I was showing were expressed in structures that are derived from the neural crest. So if there's a certain pattern to that. It's true, it was for these ones, but I mean, we looked at only at these four basically, and I mean, we didn't look at, what we found is that when we look at all 300 of them just at the positioning, because I mean, obviously they don't know if it's really controlling the neighboring gene, but let's assume it would control the neighboring gene. There was actually an increase in genes that have a role in neuronal development. So not neural crest, but neuronal development per se. And so, but we didn't follow up on this more because we actually realized that the construct that we're using has some background in the brain, so we couldn't use that for these structures. But it's true that it might be related to the neural crest, and we haven't followed up too much on these ones. But
so a good bit's known about the developmental biology of eye development in the vertebrate embryo. Can you tell me just what goes wrong in that stem?
Yeah, so it's so they are born with an eye, but it's a little bit smaller, and then the eye basically degenerates, and it's coming from the lens. So interestingly enough, if you take a lens from a surface fish and place it just a little, I mean, you can remove it with a very fine needle. You can remove it, put it onto a cavefish lens. So remove also the cavefish lens first, put it onto the eye, and then you get a cavefish which has a huge eye. So it's really also only signals that are in the lens that are playing this, and then you can do vice versa, and you get a surface fish with a HSP90 per se has nothing to do with that, other than that it does somehow regulate or keeps genes quiet that play a role in this development. We don't know. That's the excellent question, and this would be the next step to actually find the genes that HSP90 is controlling, and we're doing this. So HSP90 is usually these genes that it's canalizing, that it's keeping basically in place, are usually, or its candidates for interaction are usually kinases. So we are looking now at all the QTL intervals that we have for eye development, and look if in these QTLs there are genes that are known interactors of HSP90, and so we can screen now for all these hundreds of genes in the interval. There are ways to do that high throughput, and so we want to obviously find the actual genetic basis of what I described, and we have no idea, to be honest. So, but this is basically the next step. Yes. Yes.
And so circadian rhythm is intimately linked to metabolism.
Yes.
And these fish live in the dark.
Yes.
So have you seen anything going on with circadian rhythm?
Yes. So so these cavefish in the lab, for example, there's been shown that they still have some circadian rhythm. It's, however, a little bit disrupted. It's no longer as strong as in surface fish. In the wild, they seem not to have a circadian rhythm anymore, because probably in the lab you still feed them at the same time or something like that. So there is some Zeitgeber basically that they can use. And there is a study that recently that showed that some of this starvation resistance is because they have lost the metabolic circadian rhythm, and that saves them some energy basically. But this is just contributing to a little bit. I think the biggest reason is because they are so fat, and fat is really a very efficient way of storing your energy, and that's, I think, what's going on.
The eye development thing you just described is very strange. Is it that the crystallins in some way might aggregate when they're faced with this low conductivity environment? If you control the conductivity of the cavefish, does it save the eyes of the cavefish at all?
No, I mean it's really basic. What we did show is that if you have this low conductivity, you get this environmental stress, and then activates HSP90. So HSP90 is really busy with but of what the genes actually it is that is controlling, and what the variation is we don't know yet. But it could be crystallins, yes, of course.
Yeah, so why else would the lens be an especially vulnerable site for establishing deterioration?
No, I mean, I think we just got basically lucky that it was, because it seems like a lot of these candidates for HSP90 are genes that play a role in eye development. These interactors that HSP90 is thinking, because in many other systems, like in the fly also, you get eye phenotypes when you block HSP90. So it seems like a lot of these candidates are actually for eye development, and it's just coincidental that in cavefish, that is an important trait during cave evolution. So I think that was just coincidental that eye phenotypes are very easy to absorb, but observe. But it doesn't mean that it's only affecting eye development. It could be other phenotypes as well, like the neuromast or something like that. But it's just harder to quantify if you have 350 neuromasts versus 250. While in the eye, you can really exactly measure the orbit space. It was really very, very specific that you could see this difference.
The program deterioration structure you've already made is actually an interesting phenomenon. That's the only
Yeah, no, it's true. It could be linked to apoptosis, yeah.
There must be blind cavefish in other environments.
Yes.
Is it always the same? Is it the same mutation, or are there independent ways that you understand?
That's the problem. We don't really know what the genes are we still don't know the genetic basis of this degeneration of the eye. So if you, for example, cross different populations together, so if you cross, let's say, Pachan with Tinaja, you still get a fish that has no eyes, but they're a little bit larger already. So there is some overlap of the genes, but also some different genes playing a role. But yeah, we simply,
We were wondering if this lens.
we don't.
You might lose the eye for a different reason in a different.
It's in all the Astyanax mexicanus, it's always the lens degeneration. At least there are 30 different populations. I don't think people have looked in every population, but at least in the 3 that they looked, it's always the same developmental program for these cavefish. However, there are other cavefish, Somalian cavefish, and they have a little bit of a different program. So it's not, people argue that it's not this degeneration of the lens. And then, I think it's kind of interesting that in all cave species, this is a general trend. You always lose the eyes, you always lose the pigmentation. And technically, if there would be no selective pressure, you could just also get dog cavefish, but you never get them. So.
Maybe I miss it. Do you guys grow it in the dark or?
Yeah, we grow them in the light, the cavefish, and it does not affect. So we did the same experiment in the light and in the dark. It doesn't affect it within. We would probably need to grow them for 1,000 generations in the light to actually regain an eye, which is actually possible. There's one cave which collapsed, and cavefish actually regrow or regained an eye, but they regained it through a different developmental mechanism. It looks slightly different how the eye is shaped and everything. That's why we know it's not some surface fish that came back, but it's possible.
Dr. Davidson, last question.
Yeah.
Maybe I don't remember right, but somewhere in the back of my head I recall that Bill Jeffries also working with Astyanax.
Yeah.
in Mexico.
Yes.
He showed that there was an alteration in hedgehog expression during early
Yes.
development.
Yes.
Remember that correctly?
Yeah, that's correct.
Consistent with your comment that they're born with normal eyes and they can generate. Because he showed that there's a problem with eye development.
But it's really just, so they are born with eyes, but the eyes are slightly smaller to begin with. And that's what he's arguing, that there's sonic hedgehog is expanding.
Positions the eyes.
Sorry, say it again?
It positions the eyes, if I recall correctly.
No, I think how I understood it is just the smaller eyes, basically.
That's the orbits.
Yeah. But I think it's not, it's also, sonic hedgehog does not map to any QTL for eye development, so it cannot be a sonic hedgehog anyway directly. It must be anything upstream or something like that. But we don't know yet. The answer is, short answer, we simply don't know what the genes are. On top of it also, yes. But if it would be just have a tiny effect to that, yes.